photograph I borrowed from the Orto Fruticolo del Mugello, and I'd like to say that's me and Paula in retirement. Nice. You didn't know that.
That's maybe your idea.
Yeah, that's us. That's us with our little smokehouse there doing dried chestnuts. Of course, you know chestnuts are our staple food, and that would be the dried form. Of course, we eat chestnuts. I did for about 15 years, we had a cultivar trial in Chattanooga that we ran. It was a replicated trial. We had 20 trees of every cultivar that we could get. We had about 40 kinds from all over the world. Most of them were worthless, but some of them were really, really good. I just wanted to show one picture result from that. Based on some criticism I had of an earlier talk, I wanted to start with this one slide. This is an example of the kind of work we were doing. Pain, the cultivar Pain was one we really, really liked. It's kind of a dwarf tree for us. It was one of the smaller trees, not very vigorous, but the nuts were medium to large for the Chinese. We've never encountered the blossom end rot in this one in Chattanooga, where we did in some of the other cultivars. Anyway, that was an example of one that we really liked. This was a selection by Mike Nave from one of the Byron, from one of Jerry Payne's orchards in Georgia. But that was an interesting one. So today I'm going to talk about biodiversity and conservation, and I'm going to start with Alabama. If you haven't been to Alabama, it's worth a trip. It's a fantastically biodiverse state. We're talking about rare and endemic plants. It has a very, very high level of endemism, which means, in the context here, species that occur there that occur nowhere else. Not just plants, but also mollusks, arthropods, fungi. It's a fascinating place. It's still largely unexplored. It's a fascinating place. And so we've been very fortunate. We live in Chattanooga. We're close to Alabama. We can make excursions down there. This is the photograph here. That's Cheaha Mountain, which is the highest point in Alabama. And it's actually an outlier of the Blue Ridge. So from a geological point of view, it's the southernmost point of the Blue Ridge Mountains, which is curious because it's south of the Cumberland Plateau. So geologically, very, very different orogeny and curious mixture of different plants. This is where we found the Alabama chinkapins. Just to take a step back, we know there are some number of species of chestnuts in the world. I don't know the number. The authors of this paper, which by the way included me, listed one species in Europe, 4 species in East Asia, and 2 species in Eastern North America, one of which is divided into 2 varieties, Ozarkensis and Pumila. So, we were basing the list of species on what we thought was the most up-to-date treatment from the botanical literature. And so the division of Castanea in North America into 2 species and one species divided into 2 varieties was based on George Johnson's 1988 paper, which was the last actual revision of the genus for North America. There's a lot of controversy in the literature if you look for the number of species. For instance, the Flora of North America, which purports to be an authoritative source, lists 3 species. So they have elevated Ozarkensis to species level and Allegheny chinkapin to species level. By the way, where is Allegheny? Yeah, like who decided it was going to be called Allegheny chinkapin? By the way, who gets to decide that? By the way, I'm gonna argue that we need more work on the chinkapins. Most of the diversity is in the south and in the southeast. In particular, the range of Castanea goes all the way to Texas. By the way, we don't think about chestnuts in Texas, but they do occur there. Castanea occurs in Texas, all the way along the Gulf Coast of Florida. The species you're familiar with include what we call the American chestnut, Castanea delicata. We've got an example of an Ozark chinkapin here. Now that's a very old herbarium specimen from our work with the herbarium records. The one photograph there is at Pinnacle Mountain, which is near Little Rock, which is right at the very edge of the occurrence of Ozarkensis. And then I've got a few examples of pumila here. I have one from Florida, the lower left. And the populations in Florida are particularly interesting because they grow as a creeping shrub. And they bloom on the current season wood. They get about 2 feet tall, maybe 4 feet tall, and they form large genets. A genet, remember Sandy told us about the ortet and the ramet, but she didn't tell us about the genet. A genet is essentially a clonal population of plants that arise from underground stems, in this case, and they form large thickets. We have photographs of thickets that were maybe 50 or 100 feet across, genetically distinct from the next one, you could tell, because one was 2 feet tall, the next one was 3 feet tall. Fascinating. In the interior uplands, we see taller forms. They can be 10 or 12 or even 15 feet tall in the interior uplands. My colleague Taylor Perkins put this together. Of course, it's a trick slide. Some of these are chinkapins and some of them are chestnuts. It's not always easy to tell them in the field. It takes a suite of characteristics, looking at leaf and twig characters and stipules. Sometimes you need a microscope to look at glands and hairs on the leaves. In the absence of flower parts, they can be difficult to tell. So the flowers are the botanical difference between the chinkapins and the American chestnut is the number of pistils per cupule. And remember that the cupule is homologous to the cup in an acorn. Oaks typically have 1 pistil per cupule, so you get 1 nut per cup. Beech trees, typically you have 2 pistils per cupule, you get 2 beech nuts in every little burr. In Castanea dentata, there's 3 pistillate flowers, 3 separate flowers per cupule, so you get 3 nuts per burr. In the chinkapins, typically, but not always, we get just 1 pistil per cupule, so you get 1 nut per burr. The burr opens along a single suture into 2 valves. So those are the floral characteristics, but in the absence of flowering, you cannot tell. And so we've been fooled. In fact, in the area where we live, there are things that look like American chestnuts that turn out to be not American chestnuts. We see them as single stem trees. Here's a snag, a dead one. They have no stellate trichomes. Am I doing okay?
You're fine.
He's adjusting the volume on me. If I look to my right, it's louder. If I look to the left, it's not so loud. You can do like this.
That's better.
Like this, you like that better? Thanks. The stellate trichomes, I said that offhand yesterday, and somebody said, well, what's a stellate trichome? Those are the tiny microscopic hairs that occur on the surfaces of leaves of some species. Lots of plants have them, but essentially they're a little tuft of hairs. And they come in the closer you look, you see more and more differences. But basically, American chestnuts do not have them. Chinese chestnuts do, chinkapins do, some of the other Asian species do. The Alabama chinkapins never do. We have looked at dozens of specimens, hundreds of specimens, including those in the herbarium. We never see stellate trichomes on the Alabama chinkapins. So the lower leaf surface is hairless. It is whitish. We call that glabrous because it has a glaucous, the glaucous, they have a whitish wax on the bottom of the leaf that makes them look whitish. And of course they have the 1 nut per burr. The burr opens into 2 valves. The herbarium specimen here, is that my laser pointer? The herbarium specimen, this was Ash's 1924 collection from Lawrence County, Alabama, which was the type specimen for the species he named in 1925. He described it as a species. When we first saw these plants, and it was because of volunteers of the American Chestnut Foundation that I became aware of these. We had folks out in the field scouting for blooming American chestnuts. We're working with the American Chestnut Foundation on the backcross breeding. We had ordered pollen from Fred Hebert at Meadowview. They had found the trees and had bagged the trees. And so they had carried ladders out into the wilderness. So I drove down there with the pollen, and I climbed up on the ladder, and I took the bag off, and I see 1 pistil. And I'm on the top of the ladder and I say, hey guys, I think these are chinkapins. And they're like, who is this guy? Like, who does he think he is? So these were the American chestnuts that they had found out in the wilderness that they were ready to breed. And I'm like, no, they're not. And so we came up with a whole bunch of different ideas about them. You know, everybody had an opinion about what they might be. They could have been hybrids. If you read in the literature, there are descriptions of hybrid taxa that exhibit both characteristics of chinkapins and American chestnuts. And they have been named, and we actually wrote a paper together with the grad student, Megan Binkley, where we came right out and said we thought these were examples of Castanea ex neglecta. They matched the descriptions that Johnson used in his 1988 revision of the genus. So that's kind of what we thought they were. We used chloroplast genomes to help separate them. And I think when we started, we had three loci. When we went back and got the paper, we added 6 more loci. So the paper was published, we had 9 loci from the chloroplast genome. We were able to separate them. And what we found was that there was chloroplast sharing between American chestnut and the chinkapin taxa. And so if you know about how chloroplasts are inherited, they're like mitochondria. In plants, usually, typically, you get all your chloroplasts from your mother. And so they're a good indicator. They ought to be separated by species, right? And so the definition of the species is you breed with other members of your species, and so you're supposed to have all the same kind of chloroplast. So evidence of hybridity then, it made logical sense. The problem is, because they don't recombine, you can't really do a phylogeny using chloroplasts. They're a lousy, you can't get enough information from chloroplast genomes to study evolution and relationships. We needed a whole genome sequence. So we needed to wait for the technology to evolve and another grant. Meanwhile, I mentioned that Ash had described it as a species. It was another graduate student of mine, Taylor Perkins, who recognized that the species that we had called neglecta matched exactly Ash's description from 25. So we went back and looked at them again, and it turns out that Johnson in '88 had seen them and had annotated all of Ash's herbarium specimens. And he decided that they were Ozark chinkapins. So he synonymized alabamensis with Ozarkensis. And he went to look for them and couldn't find them and just declared them extirpated. He couldn't find them, they didn't exist. And so that was the thing, right? And so they were gone from nature. They were considered an extinct eastern disjunct of Ozarkensis. So we used whole genome sequencing. The technology improved vastly. Now we could afford it. And we used single nucleotide polymorphisms. We call them SNPs. And we can get information from these SNPs about relationships, and we can generate evolutionary, let's call them a genealogy. We can look at, we call this a phylogram. We can see relationships among the plants. And so this hypothesis would be that individual 1 and 2 are more closely related to one another than they are to individual 3. And we can apply this to the whole genus or to the whole family or to all of life. So we went back to the original samples from the 2012 study. We went back to the field and collected some more. And what we found based on this is a phylogram based on SNPs. We found that the new data support Ash's hypothesis that alabamensis is a thing. And I'm going to call it a thing. So we don't have to argue about whether it's a species or a variety, but it's definitely a thing. In other words, it's a separate clade from the Allegheny chinkapin, which is Pumila variety Pumila. It's separate from American chestnut, clearly, distinctly. And by the way, it's to scale. The length of the internodes represent the number of mutations between individuals. Okay, and so the length, you can get a measure, it's to scale the measure of genetic diversity by the length of the lines. So in the lower left down here, I have some outgroups. I've got Japanese chestnuts. I've got some Chinese chestnuts. The 2 asterisks there, those are American Chestnut Foundation F1 hybrids. Those are American Chinese F1 hybrids, and gratifyingly, they showed up right where we would have expected. And I used the verb tense would have expected because actually they were mistakes. And we found out later they were F1 hybrids. We were worried about publishing this paper because when we submitted the manuscript, we only had 2 Ozark chinkapins, which was not enough. And one of them turned out to be a hybrid. So, whoops, the little star there, that turned out to be a Chinese chestnut Ozark chinkapin hybrid. And when we went back and looked at the records, it was sent to us by a park ranger, and it was planted in the parking lot of the visitor center at the state park, which is a dumb place to collect a specimen. In hindsight, you probably wouldn't collect that one, right? So we only had 1 Ozark, and so we still had a few questions. We still had a few questions about these things. We wanted to research them further. So Taylor Perkins was pretty good at grant proposal writing, wrote a proposal to the American Chestnut Founding, and the way he pitched it was, you know, you got to make it sound like it's about American chestnut, right? Instead of about chinkapins. And so his research question was, is this hybridization? Well, first of all, is there evidence of hybridity? Can we use the whole genome sequences to see if they're hybrids? And if so, is there any adaptive value? Are there genes moving from the chinkapin species into the American chestnut that might have some adaptive value? And so that was an interesting question. The Chestnut Foundation thought so too and was willing to fund the work. It's mostly at this point, it's bioinformatics, and so it's all statistics. We also wanted to know if the Alabama chinkapins are really Ozark chinkapins. We didn't think they were. They don't look like Ozark chinkapins. First of all, they have no stellate trichomes. Ozark chinkapins are covered up with stellate trichomes, so they're very fuzzy on the bottom of the leaf. So we went back to the field. We made 4 collecting trips to the Ozark uplands. In the first of the first two collecting trips, we went in the summer to collect herbarium vouchers. By the way, I'm going to take it aside. I'm going to step over here just to make this aside. I'm an advocate of the herbarium voucher. You know, they make us deposit the DNA sequences in a repository of DNA sequences. I want to see the plant. So if you're going to publish the genomics paper, you got to have a physical plant specimen to go with it, because I want to be able to look at that, and a year from now, 10 years from now, or 50 or 100 years from now, those vouchers will be of critical importance. So we had to go in the summer when the trees were fully leafed out, preferably with flowers on them, because we want to see pistils. So we had to go in July to Arkansas and Missouri to collect the herbarium specimens, but the DNA tissue sampling is best done in March and April when the buds are just breaking, because we need young, tender leaves to get a good DNA extraction. Chestnut leaves are full of tannins and other nasty, gunky brown stuff that oxidizes when you grind it up, and so it's difficult to get a good DNA extraction from mature leaves. So it meant 4 trips. On trip 3 and 4, we did the Ouachita Mountains, which are here, which we predicted might be a transition zone. And then the southern Arkansas population that's coastal plain. So it might seem weird to you if you haven't thought about it before, but the coastal plain extends all the way to Little Rock. That's the Mississippi Delta. From a floristic point of view, it is coastal plain. And so we collected all across the coastal plain. We missed Texas and Louisiana, but we got Florida, we got South Carolina, we got as far north as Maryland. The specimens of Alabamensis, I don't know how well the color is showing up. Alabamensis has a tiny area of endemism right here, northwest Georgia, northern Alabama. And then the green dots are American chestnuts we included in our study all the way up to Maine. The sequencing work was done by Alexander Koch at Virginia Tech, and we ended up with 5 million SNPs. And I just wanted to mention that the 2012 paper where we looked at the chloroplast genome, here's the quiz question. How many loci did we have? We had 9. We started with 3, but the paper was rejected. So we went back and we did 6 more loci. We had 9 total. We got it published. And in those few years, between 2012 and and nowadays, this was 2017, 2018, the technology improved that for about the same amount of effort. Actually, it was a little bit more effort. But we have 5,769,022 loci. So it's a very large data set, which means you can't run the program on your laptop. You need some high-performance computing power. And that's where Taylor's pretty good. So here's the phylogram. These are the results of that analysis. You can see alabamensis is a thing. So I'll use the phrase clade, which means that all of these individuals, according to this hypothesis, all of these individuals are descended from that one. And in this iteration, they're sister to Pumila, but they're clearly distinct from Ozarkensis. So that debunks Johnson's claim that they're an eastern disjunct of Ozarkensis. They're not. They're definitely a thing. Curiously, there's a lot of genetic structure in dentata. So we look over here at the dentata part of the cladogram. You can see that there's interesting genetic structure. There are several major clades. And this is, again, one of those gratifying moments when your own results corroborate the results of other researchers. This pattern of genetic structure has been observed by other researchers using similar but not the same methodologies. And so we can more or less divide the structure of the American chestnut genetic structure into a northeastern, middle, and southwestern population. I'm talking about southwestern part of the range. In other words, the southernmost part of the range is distinctly different genetically than the middle and the northern part of the range. There's also an interesting observation here that was shown by Gehling and Nelson, is that if you draw an axis from the southwest to the northeast, there's also a gradient of diversity perpendicular to that, with areas east of that line, in other words, towards the coastal plain, have generally less diversity, and areas west of that line or north of that line, sounds a little bit contradictory, have more genetic diversity. We can talk about that more later if you want to. We used another analysis that we call admixture, which asks the question, what proportion of the genome at a statistical level looks like one or the other species? So in these are imaginary data here, just for illustrative purposes. If I tell the program that's a chinkapin and that's an American chestnut, if it was an F1 hybrid, it would be 50-50 Chinese, 50-50 American chinkapin. If it was a first back cross, it would be 3/4 American and 1/4 chinkapin. And so the results of our study, when we tell it, we told admixture, we said, okay, what's the structure here? There's two main clades. And we know that already, but this is a corroboration of that. We've got American chestnuts and we've got chinkapin. And what we see here is no evidence of hybridization. The little colors, the little blips of color here are probably artifacts of the software. We set the threshold arbitrarily at 10%, but that's typical for if you look in the literature. And what the explanation of this from a biological point of view is that these are probably rare alleles in the population, and the computer thinks, well, that's not American chestnut, it must be the other thing, right? And so it colored it the other way. Similarly, these little blips of color within alabamensis, within Pumila, and within Ozarkensis don't mean that there's hybrids. And we've done further analyses of these same data, and we don't really see evidence of hybridization. I'm going to go back. If I take the dentata portion of this and turn it sideways, just to make it easier to look at, I've got them arranged, in this case, I've got them arranged by latitude, the individuals from the northernmost section to the southernmost, and it shows pretty clearly that we have these three subpopulations, a northern, a middle, and a southern population. There are a few interesting things to look at here. For instance, Indiana. Indiana has members of all three of the subpopulations in the Indiana trees. And that can be explained, I think, by that axis of diversity that I talked about. We're talking about a post-Pleistocene. I wish the Holocene, the palynology guy was here today, Albert. We're talking about post-Pleistocene migrations from genetic refugia, from glacial refugia, but Tennessee, very diverse, Alabama, very diverse. If we look at the chinkapins, we've got three things. But here we do see introgression. When I use the word introgression, what I mean is evidence in the genome of hybridization. Now, it could be an artifact of some very ancient diversity, or it could mean that there's ongoing hybridization, still today. So we can test that in other ways, and we're working on other analyses of that. But for today, I'm not going to show those data. I'm just going to say that we think there's hybridization. These are introgressed individuals. And again, within each species here, I have them arranged, in this case, by longitude. And so from west to east across the thing, notice that the coastal plain chinkapins occur in the west. Those are the Arkansas coastal plain trees. These are the South Carolina, Georgia, Florida, Alabama coastal plain trees. What's interesting here is that there's a clinal variation in introgression from the south to the north, from Florida to Maryland, where by the time we get to Maryland, essentially only the interior upland form is present. I think this is worthy of further investigation. I'm very curious to go back to Florida, and of course we don't have Texas. Texas is missing completely. And then there's alabamensis. Alabamensis is over there. It's a thing. We do see evidence of introgression. And there are specimens that co-occur with pumila in some areas of the Cumberland Plateau, and we have seen them growing together with pumila, and we can tell them apart in the field. The herbarium specimens, we can tell them apart in the herbarium using historical specimens, and we also see some evidence of introgression with coastal plain and interior upland. So this lighter purple here I'm calling the interior upland form of the chinkapin. It's gratifying. Did I use the word gratifying? When you do a test and the results make sense. In other words, I'm happy because I can explain it when I see the result. But the principal component analysis is sort of some abstract math. It plots the genetic differences. Remember, this is based on 5 million SNPs in axes of difference. And so the first component separates them into 3 groups, and the second component sort of spreads the group out in 2 dimensions. And what's gratifying here is that the color codes are based on our field diagnosis. I mean, what did it look like? That's a morphotype. It looked like alabamensis. So the gratifying thing is that even in the principal component analysis, they separated clearly into 3 groups. I've got regular old chinkapins, I've got Ozark chinkapins, and I've got Alabama chinkapins. What's curious also about this is that I kind of looked at it in this, imagine it was some colliding galaxies, and these tails are being dragged out by the gravitational pull of the supermassive black hole. Never mind. These tails here, when we went back, we know the name of the individual. Behind this, there's a lot of data. I can click on that dot, I know which plant that is. Those are the plants that have greater than 10% hybridity in the admixture plot, in the structure plot. So those are the hybrids. And so we know, for instance, that these Ozark chinkapins are the ones from Pinnacle Mountain at the southeasternmost point of the range, where if you stand on Pinnacle Mountain, you can see the coastal plain. And that these pumulas here are the Arkansas coastal plain trees, where if you stand on a clear day and look, you can see Pinnacle Mountain. So they're literally within line of sight distance. Okay, and so that's gratifying. The conclusion, have the chinkapin taxa contributed to genetic diversity in American chestnut? No. They didn't. So no evidence of hybridization. Now, going back to the chloroplast study, we have whole genome sequences now. The whole genome sequence includes all of the chloroplast DNA and the mitochondrial DNA. So we've got a project underway now to go back and look at that, because we still haven't got a good explanation for chloroplast sharing. Okay, and so it may be that the chloroplast sharing is an artifact of some ancient diversity. You know, in the ancestral population, millions of years ago, there may have been a bunch of chloroplast types, and as the species differentiated into their modern, types, there was incomplete sorting of the chloroplasts. That could be one chloroplast type. Whereas in Alabama, we have 23 different chloroplast types. Okay, so it's a very, very ancient population. That is a relic of a pre-glacial population. Those are very, very old. And so we've accumulated diversity, and it's a relic of some very ancient diversity. Is Castanea alabamensis an eastern disjunct of Ozark chinkapin as claimed by Johnson? No, it's not. It's a thing. Okay? And so our conclusion is that we have genomic evidence for 4 Castanea species. Now the fact that they introgress along their margins should only bother you if you worry about that sort of thing. Do you worry about introgression along species margins? Maybe you do, right? And so, but they are clearly distinct. You know, we're not going to talk about oaks today, but if you go to the oak genomics, I mean, they literally fight and shout at each other about this kind of thing, right? And so we're in a good situation. So here's a few pictures of alabamensis just to show you what it looks like. Now the one that's illustrated here has kind of a funky leaf edge, which is sort of wavy, but I'm going to tell you that we see wavy leaf margins on American chestnut also. And if you tell them I told you, we also see stellate trichomes occasionally on American chestnuts. And I remember, this looks like a mature crowd, I remember years ago I asked Fred Hebert about that. I said, Fred, Sandy's laughing, I said, Fred, how many stellate trichomes did it have to have before it's not an American chestnut? And Fred said, oh, fuck. Right? And so I showed him the specimen, and he said that trichome rubbed off of the other one on the other branch. I'm like, no, it's stuck on there. And he was going to like rub it off. So anyway, one of the the characters that we use in the field, they are glaucous leaved on the lower surface. They have a whitish bloom. They look whitish, but there are no stellate trichomes on the leaves. They have vein hairs, and you can see a few vein hairs in this picture. The vein hairs are short and erect. The vein hairs on American chestnut are longer and they lie down. They're adpressed. But very curiously, and in every case we see them, they have what we call ciliate leaf margins. And I think you can see, the quality of the photo may not be that good, but they have trichomes, simple trichomes, which look like the vein. They look like the vein hairs right along the edge of the leaf. We never see that in American chestnut. So I already got the how many minutes left sign.
You got about 4 minutes.
But we're just getting to the beginning of my talk. This is where the talk actually starts. That was just the intro part about the chinkapin. Because the title of the talk was Conservation of Genetic Diversity. Basically what I want to tell you is that, and I said this yesterday, I think, in the group, we had the conversation yesterday about chestnut, that biodiversity is important to conserve. It's our moral, we have a moral obligation to defend biodiversity and to conserve it. So part of that is learning about it. We have to discover it, we have to understand it, we have to characterize it before we can save it. So we do look for chestnuts. You know, the caption to this one on the left is we look for chestnuts like the FBI looks for dead bodies. You know, we go through the field. Nowadays we use GPS. Occasionally we see large surviving chestnuts. We see evidence of hypovirulence naturally occurring in Tennessee, rarely but occasionally. To remind you what hypovirulence is, the fungus got sick, so there's a virus that attacks the fungus, that weakens the fungus, right? Makes it hypovirulent. Was first discovered in Italy. It was introduced into France and into the United States. But we also see it naturally occurring in some parts of the United States. The best evidence of hypovirulence as a biocontrol, I think, is at the Lasagne State Forest. This is work that was done by Gary Griffin from Virginia Tech and his coworkers. These were scions that were taken from large surviving trees, grafted onto some wild-type sprouts in an experimental forest, and then inoculated with an Italian hypovirulent strain. The controls were not grafted wild types, and scion taken from wild type trees. So this shows two different things. One, it shows that hypovirulence can be used as a biocontrol. It also shows that all American chestnuts are not uniformly susceptible to blight. There is variation in blight susceptibility within the species. And the fact that all of the wild type grafts eventually succumb to blight, despite being inoculated, is good evidence to support this idea that these large surviving Americans, and when we use the term LSA, we consider a tree that's about 10 inches in diameter or greater, that has blight and has had blight for many years but didn't die. Those LSAs are probably special. And so this is evidence to that. And by the way, this is at the Lasagne State Forest in Nelson County, Virginia. It's worth a trip down there to see these trees. The problem for Gary was he only had 2. There's only 2. The reviewers don't like that. Incorporating genetic diversity, this is the conservation part. It takes labor, it takes effort, it's resource intensive. So here's an example from a real work that some of my students did. You scout the trees, you collect scion in the winter, we graft, we grow them in the greenhouse, we use highlight. I've got 3 minutes to force blossoms. I didn't show you that picture, but we've got some big bright lights. If you want to know about lighting, the guy said the other day, talk to the pot growers. They know a lot about lighting, right? We took the pollen into the field, we made the crosses, and we planted the seedlings into the conservation orchard. So these are orchards that were designed as ex situ conservation. The advantage here is that if they do bloom, I've captured those genes as hybrids. I'm going to skip the part about grafting because I think everybody here, if you haven't learned about grafting, go to the grafting workshop. I've taught grafting many times. I'm just also going to say that no one that I have taught how to graft didn't cut themselves. The grafted trees we can grow in containers. They become little bone size. I consider this a stopgap. This is not long-term storage. This is not a good option for genetic repository. But because we can force these into bloom, the magic combination of sunlight, fertilizer, and water, I can get them to bloom. And I can have in containers in a short row, I've got dozens or hundreds of genotypes. That saves hundreds or thousands of miles of driving in the car, climbing up ladders in the wilderness. I can collect pollen directly from those trees and use them in crosses that we have. Just to remind everybody, I'm really out of time, but we're breeding for 2 diseases in the southeast. We also have Phytophthora root rot. Just to remind you, it's a pathogen caused by an oomycota, an oomycete. It's not a true fungus, a fungus-like microorganism. We've done a lot of work using Tennessee isolates from symptomatic orchards, and the definition of a symptomatic orchard is all the trees are dead, right? They all died. They don't resprout from the base. I think of it as curriculum. These are my students doing the inoculation. Aren't they, I won't use the word, aren't they attractive? The results are dramatic. Most of the trees die. We have these killing tubs. We put them in there. We inoculate the tub with Phytophthora. The trees die. The American chestnuts are deathly allergic to Phytophthora. In 3 weeks, they're showing symptoms. In a month, in 6 weeks, they're dead. They have no living roots. Sometimes the tops don't die right away. We take the survivors from the tub and plant them into the orchard. In this particular experiment, these were what we call better B1s, BB1s, where it's a first back cross. I've got an F1 hybrid that we know has Phytophthora resistance. It's a Chinese-American hybrid. And we cross it to an American. But in this case, the American tree was a selected 3rd or 4th back cross tree from the American Chestnut Foundation's breeding program. So it's a American type that has some level of blight resistance that it inherited from its Chinese ancestor. We call these better B1s. Some of the better B1s are growing really, really well. They've got good American form, and they're planted back into the symptomatic orchards. In other words, these were replanted into an orchard in which all the trees had died. And so they're growing very well. They've been through the killing tub, and now we're evaluating them for their growth potential. We're also breeding for blight resistance. I'm going to skip this part because I think we've covered it in other contexts. If you don't know about the backcross breeding program, I do want to mention that it's a wonderful example of community science. It's citizen science. We have thousands of volunteers in the American Chestnut Foundation who are conducting the breeding work. They're growing the trees on their own land. They're dedicating their own resources to doing this. We have hundreds of thousands of trees that we planted from Maine to Georgia and Alabama. It's a fantastic example of citizen science and community science. I just wanted to mention that. A couple of pictures of the nursery because I'm really proud. That's my son. When the trees grow up, we try to kill them. And so to make progress, as a plant breeder, you know this, the selection process, you have to do it rigorously and carefully. In other words, I have to be able to kill the trees to be able to make selections for the resistance. It takes a lot of resources. It takes about 10 years just to answer the question, do they have resistance? We're trying some alternative methods. Small stem assays, by the way, that was a really good tree. We rogue in the backcross level, a straight backcross F1, it's about 1 out of 8 is selected. At the F2 level, it's closer to 1 out of 100 because we're really looking for just the elite trees. Of course, we've learned in the last 40 years that it is not simply inherited at a few loci. There are dozens of loci, maybe hundreds of loci, but we are making gains, and we can make gains that are measurable at every generation. And we're using genomic tools now to help us make selections. One of the alternative methods that we're using for screening is what we call a small stem assay. We can cut the tip off, or we can inoculate the twigs. The problem with this is not a very high resolution. I can separate Chinese levels from American levels of resistance, but I cannot separate intermediate levels of resistance. And so at the backcross level, I can't make selections within a family. But I can compare families to one another. And we use it for progeny testing. We've had pretty good luck. Just to remind you, it's very harsh. That's the Chinese control. A tree that size can be easily killed by chestnut blight. Even the most resistant varieties can be killed by chestnut blight if you inoculate them. So it's difficult. I think the snipped tip method is a little better because it's less destructive. It already stimulates the tree to produce side branches, and so it's less destructive of the tree. If you want to keep that tree for another purpose, you can keep growing the tree. That was developed by Marty Cipollini at Berry College, and we're using that snip tip method. We also thought we could get better resolution if we used the hypervirulent strain. It was a great project. That's Trish Wynn. She got interesting results. I'm going to skip the results really and just tell you that it didn't really work the way we wanted it to. But what we did show is that there's a very good correlation between the proportion of American in the mother's ancestry and the blight resistance in the progeny, which supports the hypothesis that blight resistance is polygenic. So many genes. I had another student who did a larger assay this year ago, Uma Plambeck, more than 1,000 seedlings in her trial, and Jared Westbrook did some statistics on there, and here's a correlation between the long-term phenotyping of the mothers. So for the mothers in this project, we had 1,000 seedlings, but we knew a lot about the moms. We have whole genome sequences for the moms. We know all about their heritage, their pedigrees. We have information about them from other progeny tests. And so, again, this is one of those gratifying moments. There was a very strong correlation here between the progeny, the average blight resistance of the progeny, and the long-term phenotype of the parents, which tells us that, well, for one thing, blight resistance is heritable. In other words, plant breeding works. It's just a long process. We haven't been able to fix resistance in the population. We probably never will get trees that are homozygous at all those loci for blight resistance. And so we're going to have to just deal with that in some other way. Curious part of it, and I know I'm over now. I'm ending. I'm getting the kill sign now. I wanted to share this with you. Uma was, we loved Uma because she was pure naive. And what I mean by that is she didn't have any expectations at all about what was going to happen. And we threw this at her. I had 8 different species. I had Henryi, Seguinii, Dentata, Alabamensis, Ozarkensis, Crenata, Sativa, and Mollissima. And these were the numbers she got. So according to Uma, Sativa was the second most resistant type after Mollissima. Okay? Crenata, it was more resistant than Crenata. Now this, you know, I like to say to my students, remember, the tree didn't read the textbook. They didn't know they were supposed to be resistant. But what was really curious over here is that Seguinii and Henryi were terrible. There are a couple ways to explain this, and we don't have to go into all the details. But the point here is, don't believe everything you read. You can do your own research if you want. We're going to do this again. Like I said, the assay was non-destructive. We have the trees. We kept them another year. I'm going to do some sequencing on these to find out if these are really sativas. We don't really know what these are. We got those from someplace in Georgia, from a botanical garden, right? So we'll learn more about this as we go, but those are curious for 1 year results, and I thought I wanted to share that with you. Just one photo to end is some of our examples of long-term phenotyping. What I mean by long-term phenotyping, these are backcross hybrids that were inoculated, some of these more than 10 years ago. They look kind of crappy, but they are alive, and they are producing nuts. Last year from our backcross orchards in Tennessee, we harvested 50,000 seeds, and we're growing them in collaboration with some of the state nurseries for some silviculture trials in western North Carolina and parts of Tennessee. And I didn't do this work all by myself. We've got external funding. I've got a lot of students that help, and these are the co-authors of the paper that we're working on at the moment. That's Taylor Perkins, me, Alex Sandercock, Jason Holliday from Virginia Tech, Ron Revord, University of Missouri, Fred Payet, a living treasure, if you haven't met Fred, Jared Westbrook, the American Chestnut Foundation, Paul Sisko. These are some of our recent papers, and I'm going to stop. I think I'm probably out of time for questions. Sorry.
No, we can do one. We can do one more. You call on them. Don't call on Sandy, though.
Okay, this gentleman right here. Yes, you.
Tim.
This is, I guess, topical adjacent, but the TACF said that they've seen several incursions of Chinese into American throughout the millennia.
I don't believe any of that stuff.
That was pollen coming from China?
No, that's bunk. That's bunk. You can tell Jared Westbrook I said that's bunk. I don't believe, no, I just don't think that's plausible. You might see a signal of Asiatic chestnut as an artifact of some very, very ancient, common ancestor. But I don't think there's pollen flow from China to North America.
I'm just thinking if it was very...
We have fossil castanea from Oregon and Washington, from Idaho. We have fossil chestnut leaves, within the last 10 to 2 million years, all throughout the southeastern United States. I'm not convinced that the center of origin of castanea is Asia anyway. We have fossil chestnuts from Greenland. We have fossil chestnuts from northern Germany, from the coal forests in the coal age forest in northern Germany. So consider that over the last many millions of years, the continents have moved. We think there may have been a westward migration from Europe into North America, and people think, oh, there's a land bridge over Greenland. No, it's not a land bridge. It's that, 40 million years ago, North America, Greenland, and Europe were the same one continent, and they had forests. So I want a question from over here. Yeah.
At this point, how do you feel about trichomes for species identity?
Well, I like trichomes. I think you should look at them. I think glandular trichomes are also important. So we're talking about simple trichomes and glandular trichomes. The problem with the glandular trichomes is you need a better microscope to see them. And you can't always see them, and they can fall off. So just because you don't see them doesn't mean they weren't there. So young, fresh leaves, early, late spring, early summer. For Japanese chestnut, for instance, the very distinctive trichome looks like scales. American chestnut, the glandular trichomes have 4 cells. Just 4 cells. On the chickapins, the glandular trichomes look like little sort of sausage-shaped things with no head. Right? So the American chestnut trichome, the glands, looks like a little mushroom. But there's 4 cells in the top. So those are helpful if you've got the microscope to see them.